Hello and welcome back once again to Biobasics. This is Dr. Komal Acharya and here I am presenting translation in prokaryotes. As this particular topic is little bit lengthy, I have divided it in two sections for better understanding. So here I am presenting the first part of the translation in prokaryotic cells. In our last video, we have seen that the genetic information stored in DNA is transferred to mRNA by the transcription process. Now, this information is further transferred from the linear polynucleotide sequence to a polypeptide chain, or we can say proteins, through the process called translation. Proteins are the functional product of the gene expression. As I told you earlier, the gene expression is a process by which the genetic information stored in DNA is converted into the functional product, that is protein. So protein synthesis is the final step of the gene expression. Now we all know that proteins are made up of 22 amino acids and each amino acid has a specific codon. But what is this codon? So we all know that in DNA, the information is stored in form of a sequence of nucleotides. A nucleotide could be adenine and thymine and guanine and cytosine. When it is converted into RNA, then the nucleotide could be adenine, guanine, cytosine and uracil. Uracil replaces the thymine in case of RNA. So, a set of three such nucleotide is known as codon. Now each codon specifies one amino acid. So the genetic information in form of codons are transferred into amino acid sequences by the process called translation, or we can say protein synthesis, which takes place on ribosomes. And the tRNAs recognize these codons which are present on the mRNA. Right, so DNA is converted into mRNA by transcription process, and this mRNA is further converted into polypeptide chain, or we can say proteins, by the process called translation or protein synthesis. But before going into detail about this process, protein synthesis process, let's talk about some important elements which are involved in the particular protein synthesis. So here are the different elements which are involved in protein synthesis: mRNA, tRNA, rRNA and ribosome. These are the main elements which are involved in particular process. Let's talk about mRNA first. mRNA is a messenger RNA. Why it is called messenger? Because it works as a messenger. It carries the genetic information from DNA to the protein synthesizing machinery of the cell, that is ribosome. It carries the information copied from the DNA in form of a sequence of triplet codons. mRNA is a polynucleotide sequence which contains sequence of triplet codons. Let's talk about types of the mRNAs. There are two types of mRNAs. First one is monocystonic mRNA. It represents only a single gene, which means after translation only one type of protein molecules will be synthesized. Whereas in case of polycystonic mRNA, the particular mRNA represents more than one genes. So after translation, more than one kind of protein molecules will be synthesized. As far as the prokaryotic cells are concerned, most of the prokaryotes has polycystonic mRNA. Now let's talk about the structural aspects of the mRNA. Basically, mRNA contains two regions: coding or translatable region and non-translatable region. In case of coding or translatable region, it has codons starting from the start codon, from where the protein synthesis begins, and a stop codon where the protein synthesis ends. Now there are always extra regions on both the ends, that is 5' and 3' ends of the mRNA, which are untranslatable. So the non-translatable region which are present upstream of the start codon is known as 5' UTR, because it is present on the 5' end of the mRNA, and UTR stands for untranslatable region. It is also known as ladder sequence or ladder region. And The non-translatable region which are present on 3' end, or we can say downstream of the stop codon, is known as 3'UTR, or we can say it trailer sequence or trailer region. Now, as I told you earlier, the prokaryotic mRNA is polycistronic in nature. So it contains multiple cistrons. Now, there is a new term, cistron. What is this cistron? Cistron is a section of DNA or RNA molecule that codes for a specific polypeptide. Right. So, an mRNA possesses more than one cistron, a polycistronic mRNA. These are different cistrons which are separated by an untranslatable region that is known as intercistronic spacer. One more important thing. Protein synthesis begins when the ribosome binds to the mRNA at particular site. This particular site is known as ribosome binding site, and an mRNA has multiple ribosome binding sites. This particular site contains a consensus sequence 5-AGG, AGG, 3-. This consensus sequence is known as the Shine-Dalgarno sequence. This is the particular site where the ribosome attaches with the mRNA. Another important thing, each mRNA contains a contiguous and non-overlapping string of codons known as open reading frame. This is the mRNA strand and this is the triplet codons. These are the triplet codons. Okay, so this is known as the open reading frame which are read out by the ribosome. Okay, now there are three options of open reading frames depending upon where the decoding starts. Okay, so if the start codon is this A, then the polynucleotide sequence will be this. If the start codon changed, means instead of A, now the start codon is G, then the polynucleotide sequence which is synthesized will be different. And if the start codon is U instead of G, then again the polynucleotide chain which will be synthesized will be different. So the mRNA can be translated in three different frames depending upon where the translation starts. Right. So this was the open reading frame of mRNA. So this was mRNA. Now let's talk about tRNA. tRNA, or we can say transfer RNA, is also known as adapter RNA. Basically, it is a single chain having 73 to 93 nucleotides that folds and form a clover leaf-like structure. This structure contains 5 different arms and loops. Let's talk about acceptor arm or acceptor stem first. Acceptor arm, the name itself suggests that it accepts the new amino acid that attached with it and converts this tRNA into a charged tRNA. This particular arm has 7 base pair of nucleotides along with 4 unpaired nucleotides. This nucleotide sequence possesses 5-CCA 3- end, which is conserved for all the tRNAs. Now, another one is D arm. This arm has 3 to 4 base pairs and a loop that contains a modified nitrogenous base dihydrouridine. Third one is anticodon arm. The name says that this arm has an anticodon having 3 nucleotides in sequence that recognize the codon which is present on the mRNA, and this is crucial for the protein synthesis process. Next one is T arm. It has a 5 base pair nucleotides containing arm and a loop which has an unusual base pseudouridine. There is another arm, variable arm, that contains 4 to 24 nucleotides which is variable in nature. But this is the primary structure of tRNA. It folds into L-shaped 3D structure through complex ternary interactions between different arms. Okay, now these unusual bases like pseudouridine and dihydrouridine present on the tRNA molecule are created post-transcriptionally. After the transcription, when the RNAs are created, several post-transcriptional modifications are there, which are basically enzymatic modifications in normal nitrogenous bases, and as a result such kind of unusual bases are created which are present on the tRNA molecule. Now each amino acid has its specific tRNA, and tRNA attaches with the amino acid at this receptor arm and carries it to the mRNA. In prokaryotes, there may be 30 to 45 different tRNAs. And as we know, there are 22 amino acids. So, some amino acid may specify more than one tRNA molecule. Okay, so this was the brief introduction of tRNA. Now let's talk about RNA, rRNA. rRNA or ribosomal RNA associates with the R proteins and form ribosome. In prokaryotic cell, there is always a 70S ribosome. Now what is this capital S stands for? So capital S stands for the Swedberg's unit. It is a unit to measure that how fast a molecule can move in a centrifuge. So particular prokaryotic cell contains 70S ribosome. The 70S ribosome contains 16S rRNA, 23S rRNA and 5S rRNA. Each has different roles. 23S rRNA has a catalytic role. It helps in peptidyl transferase activity during translation. We will talk about it later on. 16S rRNA has role in recognition. It is involved in proper positioning of mRNA on the ribosome. Now let's talk about ribosome. Ribosome is like a small migrating factory that moves along mRNA in 5' to 3' direction and catalyzes the assembly of amino acids to form a polypeptide chain. So basically ribosome is a ribonucleoprotein particle present in the cell that contains rRNA and R proteins. Ribosome is made up of small subunit and large subunit, and each subunit contains these rRNAs and R proteins. As we learnt, prokaryotes have a 70S ribosome. It is composed of 50S subunit, large subunit, and 30S small unit. 50S subunit has 5S rRNA, 23S rRNA along with the 31 to 34 R proteins. Whereas 30S subunit possesses 16S rRNA and 21 R proteins. Now, this 70S ribosome contains 3 tRNA binding sites, P site, A site and E site, which are crucial for protein synthesis process. Let's talk about them one by one. First of all, P site. P site stands for peptidyl tRNA binding site. It holds the tRNA molecule that is attached with a growing polypeptide chain. A site stands for amino acyl tRNA binding site. It holds an incoming tRNA that is charged with an incoming amino acid. And E site, exit site. The deacylated tRNA, or we can say tRNA that is not attached with the amino acid, exits through this site. Okay, so this is the structure of ribosome. But in eukaryotic cell or prokaryotic cell, in both these kinds of cell, the translation process goes on simultaneously. It means that the mRNA molecule is being translated by many ribosomes, more than one ribosomes at a time. So you can see in the structure that this is the mRNA molecule, mRNA strand, and there are multiple ribosomes over that molecule attached with it and translating more than one polypeptide chains at a time. So this particular structure is known as polyribosome or polysome. This is true for both the prokaryotic and eukaryotic cell. Okay, so these were the different major elements used in protein synthesis process. Now let's talk about protein synthesis process is divided in 3 different steps: initiation, elongation and termination. But before initiating the protein synthesis process, 2 essential processes occur. First one is activation of amino acids, or we can say charging of tRNA molecule, and another one is formylation of methionine. Let's talk about activation of amino acids first. Amino acids are immobilized, so they cannot travel by themselves to the ribosome where the protein synthesis occurs. So they need someone or something that carries them to the particular site. So here the tRNA serves as a vector that carries it to the mRNA which is attached with the ribosome. This process particularly occurs in 2 steps. In step 1, amino acid is activated by ATP molecule. And AMP is added to the amino acid and form amino acyl AMP. This particular process is catalyzed by an enzyme amino acyl tRNA synthetase. In the second step, amino acid is transferred to the 3' end of the tRNA molecule, releasing AMP. Here what happens is, you can see the tRNA molecule here. Here is the amino acid which is going to be attached with the tRNA molecule. This is the adenine. Adenine belongs to the CCA sequence from the acceptor arm of this tRNA molecule. So, the carboxyl group of the amino acid is attached with the hydroxyl group which is present on the 2' or 3' carbon of this adenine, right, of particularly pentose sugar of this adenine molecule. So this ester bond is formed between the carboxyl group of amino acid and hydroxyl group present on the 2' or 3' carbon of the pentose sugar of adenine. So this is the way the amino acid is attached with the tRNA molecule. And now the tRNA is charged tRNA. This particular bond is very energetic in nature, and that's why it is known as activation of amino acid. And here the tRNA is being attached with the amino acid. So this process is also known as charging of tRNA molecule. Now another important thing. Each amino acid has specific amino acyl tRNA synthetase enzyme. I told you earlier that each amino acid has specific tRNA. So this particular enzyme ensures that the correct tRNA is attaching with the correct amino acid molecule by identifying the particular anticodon and the acceptor arm present on the tRNA molecule. So this was the first process, essential process. Let's talk about the another one. The next essential process is formylation of methionine. In case of eukaryotic cell and prokaryotic cell as well, the start amino acid or initiating amino acid is always methionine. Why? Because the start codon always specifies the methionine. As far as the prokaryotic cell is concerned, this methionine is always formylated. Why? See, here you can see the methionine structure. This is the C terminal of the methionine and this is the N terminal of methionine. Now, it is attached with this initiating tRNA molecule. So, this is the tRNA molecule and this is the methionine amino acid attached with it through the C terminal. Okay, so this is met tRNA, we can say. Now, this particular amino acid has this free N terminal which is highly reactive in nature. So, it can react with any group, right? So, to preserve the conformation, the proper conformation of this polypeptide chain, this N terminal has to be blocked or has to be reacted with some particular group. So, here the formyl tetrahydrofolate donate its aldehyde group to the met tRNA molecule, or we can say methionine amino acid, and convert it into N-formyl met tRNA. Here you can see the N terminal is attached with this aldehyde group which is donated by formyl tetrahydrofolate molecule. This particular reaction is catalyzed by transformylase enzyme. This is true for only prokaryotic cell. Formylation of methionine occurs only in case of prokaryotic cell. Okay, so now this initiator tRNA is attached with the formylated methionine, and this is needed for the tagging purpose also, because it identifies or it indicates that yes, this is the initiating amino acid or this is the start amino acid. So this formylation of methionine occurs only in prokaryotic cell. Later on, this formyl group is also removed by deformylation process later on after the process of translation, right? So this was the pre-preparation for the protein synthesis process. In my next video lecture, I will talk about the whole protein synthesis process in detail. So if you like the video, please click the like and share button and please subscribe my video channel. Thank you very much.